So here we have a DNA molecule. This is a double-stranded helix that stores our genetic information, and I'm going to be demonstrating how it replicates. So in the initiation phase, we have an enzyme called helicase, and this uses ATP to break the hydrogen bonds of the helix and starts to unwind it. Slightly ahead of that, we have an enzyme called topoisomerase, and this essentially works ahead of this guy to prevent the supercoiling that can happen on this side of the strand due to the tension. So these work together, and these move along the DNA strand, opening it up, and that creates this part here that is called the replication fork. And the point that the helicase starts at is going to be called the origin of replication. So now we have two strands. The strand that is running from 5' to 3' towards the replication fork is now going to be our leading strand. And the one that moves from 3' to 5' again towards the replication fork, that is now became our lagging strand. Before new DNA can be built, this enzyme, called primase, needs to lay down a short piece of RNA called RNA primer. The primer gives the next enzyme a place to start. This enzyme, DNA polymerase, extends the primer by adding new DNA nucleotides in the 5' to 3' direction. On the leading strand, this can happen continuously towards the replication fork. So as this moves along, we are getting a new strand built. Now for the lagging strand, since the DNA is oriented in the opposite direction, the replication can't run as smoothly. Again, primase comes in and drops in little fragments of RNA primer. Now, the DNA polymerase builds short, separate DNA segments called Okazaki fragments, which are produced in a stop-and-go fashion away from the replication fork. So now we have little pieces of the RNA primer and the little fragments. Next, we have this little enzyme called ligase that comes in and acts almost like glue and is just going to bind these together. After the ligase has turned the Okazaki fragments into one complete continuous strand, we now have two strands that are fully synthesized and connected, which means the replication is complete. These each have one strand of the original and one newly built strand, and this is called semi-conservative replication.